Assalamu alaikum. I'm Nojesh Muhammad bin Islam. Welcome again to Learn Medical with 31. This is the second video of neuromuscular junction. In this video, I'll be describing about the transmission process of neuromuscular junction. In the previous video of neuromuscular junction, I have mentioned about the definition and other medical terms related to the neuromuscular junction. In this video, I'll be directly going through the transmission process. So let's start with the transmission process. Okay, during the transmission process, first a nerve impulse, it spreads all over the axon terminal, and this causes the opening of the voltage gated calcium channels. When the voltage gated calcium channels are open, it allows the calcium to diffuse from this synaptic space into the interior of the axon terminal or nerve terminal, and this calcium ion are believed to cause an attractive influence over these synaptic vesicles, or you may see in this figure, this calcium ion cause an attractive influence over these synaptic vesicles. So the synaptic vesicle coalesce with each other, and later these synaptic vesicles are fused with this membrane, this neuronal membrane, or you may say this one is also neuronal membrane. This one is a bigger picture of that one, a zoom in picture. So this synaptic vesicle, you see they're fusing over here, they're fusing with the neuronal membrane, and they release their neurotransmitter, the acetylcholine, by a process of exocytosis into the synaptic space. So what is happening here? First, a nerve impulse coming here into the axon terminal, then it is causing the opening of the voltage gated calcium channels, and calcium ion is diffusing from the synaptic space into the interior of the nerve terminal. Then this calcium ion are believed to cause an attractive influence over this synaptic vesicle or synaptic vesicles. Then synaptic vesicles coalesce with each other, then they fuse with the neuronal membrane over here, like this way. This is a semicircle. These vesicles are fused here, and by a process exocytosis, it is releasing its content. That's why it seem to be semicircle, because the other half portion of this circle, it is not even a circle, this is a vesicle. So as it is a two-dimensional structure, so it seems to be semicircle. So it is releasing its substances like acetylcholine into the synaptic space. Now the question is, what does this acetylcholine do over the postsynaptic muscle fiber membrane? Okay, now what does the acetylcholine do over the postsynaptic muscle fiber membrane? As we said, now the acetylcholine is in the synaptic space. This dot represents the acetylcholine. Then this acetylcholine binds with the acetylcholine receptor that is present, the acetylcholine receptor that is present on the mouth of the subneural cleft, or near the mouth of the subneural cleft. And this acetylcholine receptor is a complex protein structure, which is composed of 5 subunits of protein. Among them, 2 are alpha subunit, 1 is beta subunit, 1 is gamma, and 1 is delta subunit. When 2 molecules of acetylcholine bind with 2 alpha subunit respectively, it causes a conformational change in the structure or in the acetylcholine receptor, and this this causes the opening of the acetylcholine receptors. I'll tell you one thing. This acetylcholine gate is about 0.65 nanometer, which is sufficient enough to pass the sodium ion, and this causes the sodium ion to get diffused interior of the muscle fiber membrane. So when the sodium ion is diffusing inside of the muscle fiber membrane, it produces a local positive potential all across the muscle fiber membrane. This is known as end plate potential. This is very important. End plate potential. You may be asked during the Viva, what do you mean by end plate potential? Describe. So you will say like this. Okay. This end plate potential later on produces a action potential. Again, this end plate potential later on produces an action potential which is responsible for the contraction of muscle. So neuromuscular junction was up to here, but I would like to add some more information. Actually, these information are not for passing. They may get you some more marks. So as I have mentioned, the acetylcholine gated channel has a diameter of about 0.65 nanometer. This is large enough to pass any other like positive ions through these channels, including that potassium and magnesium. So why are we only talking about the sodium ions? Because in practice, far more sodium ions flow through that acetylcholine gated channels than any other positive ions. There are two reasons behind that. First of all, in the extracellular fluid, the sodium is concentrated in more concentration than any other positive ion. And secondly, the resting membrane potential of a large skeletal muscle fiber is about minus 80 to 90 millivolt. I think Tawhid Zaman has discussed in his video, and this minus 80 to 90 millivolts pulls the positive sodium ions inside, and simultaneously it prevents the efflux of the potassium ions. For these two reasons, sodium concentration is far more, or sodium flow is far more than any other positive ions. Now what about the negative ions? Why do not they pass? Because there is a strong negative charge that is present in the mouth, surrounding the mouth of that channel. So that negative charge repels the negative ions. Thus, the negative ion cannot pass. So that was up to here. I think you have enjoyed the video. Stay with us. Stay with Learn Medical with 31. Allah Hafiz.